Drug delivery systems are engineered technologies for the targeted drug delivery and controlled release of therapeutic agents. In targeted drug delivery system, we increase the medicine concentration in some parts of the body so that the drug will not kill any healthier cells. In targeted drug delivery system, we use drug delivery vehicles to carry the drug to specific locations of the body. Until now, we have seen how different drug vehicles like liposomes, DNA origami, magnetic nanoparticles, and gold nanoparticles are used in carrying drugs to different target sites. Now, I'm going to discuss how targeted drug delivery is achieved using carbon nanotubes. Carbon nanotubes are molecular tubes formed from rolled up sheets of graphene. They are hollow, nanometer thick cylinders made of carbon. Carbon nanotubes have good transporting capabilities combined with surface modifications and unique physiochemical properties. For cancer therapies, carbon nanotubes are good drug carriers in the targeted drug delivery systems.
Carbon nanotubes can be prepared in many different methods. Arc evaporation. Arc evaporation method is also known as electric arc discharge method. It generates the highest quality carbon nanotubes. The method consists of two graphite electrodes in the helium. A current of around 50 ampere is passed between the electrodes, causing some of the graphite from the anode to evaporate and condense on the cathode. This deposit contains the carbon nanotubes.
Laser vaporization. A pulsed laser is fired at a graphite target in an inert environment. At high temperature and pressure, the target is usually placed at one end of a 50 centimeter quartz tube. The nanotubes are collected at the opposite end.
Other methods of preparation include diffusion flame synthesis, electrolysis of graphite, hydrothermal treatment of polyethylene, explosive decomposition of picric acid, and treatment of polyesters. Carbon nanotubes can be used to create novel sensors by coating the nanotubes with specifically designed amphiphilic polymers. This approach helps to recognize different targets and monitor various diseases. The new polymer-based sensors offer a synthetic design approach, which help detect multitude of infinite targets. Due to their bonding structures, carbon-based composition and fabrication processes, the nanotubes execute key properties that help them be ideal drug delivery systems. These include increased strength, high electrical conductance, good heat resistance and dissipation. Carbon nanotubes play an essential role in chemotherapy. Doxorubicin, or DOX, is the most efficient anti-cancer drug. DOX has a drawback that it can cause death of surrounding healthy cells in the body. We can use carbon nanotubes as DOX capsules to deliver the drug to the targeted cancer tissue. How well we make the carbon nanotubes can help us determine how well it can detect the cancer cells. Carbon nanotube can be injected into the body, and the nanoparticles which carry the drugs with them can be distributed through the circulatory system of the body and delivered to the site of the tumor. The nanoparticles are so small that they can enter the leaky walls of the tumor vascular, which are formed during the process of tumor growth. And the GOX nanoparticles concentrate within the tumor tissue. The nanoparticles penetrate inside the tumor tissue as the surfaces are coated with specific cancer markers. Nanoparticles releases the DOX once they are inside the tumor cells. The direct delivery of DOX molecule will cause the cancer cell to die. This method gives us selectively release anti-cancer drugs to only targeted cancer tissues and prevent killing healthy cells. While the carbon nanotubes made using the processes as described before have impurities, not open-ended, not length-controlled, dirty, tangled carbon nanotube bundles. Traditional carbon nanotubes have traces behind sometimes heavy and toxic metals such as cadmium, lead, and aluminum. Medical grade molecular rebar solves all the disadvantages the traditional carbon nanotubes have. Drugs can be loaded inside the hollow tubes of MGMR or bonded to the exterior. Unlike traditional carbon nanotubes, MGMR is composed of individual discrete, dispersed, length controlled, surface functionalized, multi-walled carbon nanotubes. The modified carbon nanotubes have relatively long length and small diameter. Due to this fact, they can enter cells and also cross blood-brain barrier. While there is a lot of research going on carbon nanotubes as targeted drug delivery system for anti-cancer molecules, they have not been yet up to the commercial standards due to the fear of toxicity in traditional carbon nanotubes and due to the small size, large surface area, and eye aspect ratio. Small to large molecules can load as large as 10,000 daltons onto the MGMR covalently or non-covalently onto the exterior such as insulin, chlorambucil, and C5 dye.